Welcome everybody. Thank you again for joining our webinar, which is on hit lead optimization with the aid of StarDrop. So the next thing I'd like to do is introduce our speakers and panel today. We're very lucky to have two guest speakers today, and I'm also joined by my colleague Matt Siegel. Our main presenter is Dr. Claire Le Manach, who is the Principal Scientific Officer at H3 Drug and Discovery in Cape Town. Claire has a background in chemical engineering, having specialized in organic chemistry. She moved on to do a master's degree in molecular chemistry at the École Polytechnique in Palaiseau. And she then went on to do her PhD in 2008, with her research focused on identifying antitubulin compounds using dynamic combinatorial chemistry. Her first postdoctoral fellowship was at the Center for Drug Design at the University of Minnesota in Minneapolis, where she was working on antibacterial agents with the aim of identifying phosphonate derivatives active against Giardia lamblia. Claire moved on to Cape Town in 2010, where she joined Professor Kelly Chibale's group as a postdoc, beginning her research into malaria and working in collaboration with Medicines for Malaria Venture. Since February 2015, she has been the Principal Scientific Officer at H3D Drug and Discovery. And over the course of her postdoc at the University of Cape Town, she has worked on different projects related to malaria, one of which has led to the development of a potential anti-malarial agent that is currently undergoing phase 2 human trials. Last year, a backup compound also made it into the preclinical phase, so great success on those programs. She has a number of publications over the last 6 years, with these appearing in journals such as the Journal for Medicinal Chemistry, but other articles, journals such as Science Translational Medicine and the Malaria Journal. She's also an author on 2 patents. Our second guest speaker, panelist, is Dominique Besson. Dominique is a chemical engineer by training and has over 25 years of experience in the life science research and development industry. He's worked at WHO as well as various other pharma and startup companies in leadership positions. And he's fairly recently joined Medicines for Malaria Venture as the Discovery Data and Compound Manager in January 2017. And the final member of the panel is my colleague, Dr. Matthew Siegel. Matt is the CEO here at Optibrium. Matt has a background in theoretical physics, having done a PhD there at the University of Cambridge, having graduated from the University of Oxford. Matt was formerly the Senior Director responsible for Biofocus DPI's ADMET division, a company that is now owned by Charles River Laboratories. And Matt is the co-founder of Optibrium with Ed Champness when Optibrium was formed out of Biofocus back in 2009. So before handing over to Claire, I'd like to just ask if Dominique Besson would just give a short introduction to the work that MMV does and how they operate as a not-for-profit organization.
Okay, so I'll give a very short introduction to MMV. So MMV is a PDP. PDP is a Product Development Partnership, which was officially launched in 2009 to bridge the gap in new drugs for malaria. Since then, MMV is working with public and private sector to discover, develop, and facilitate the delivery of new effective and affordable drugs for malaria. We have been collaborating with UCT since a very long time, as UCT is one of our first academic partners, and starting in 2009, we've worked on various projects where MMV was playing the role of mentor and the UCT was the lead scientist. And those projects, we have been quite successful in doing that, because recently one of the projects delivered the first ever drug compounds developed on the African soil that was moving to human trials. In summary, this is it.
Super. Thank you very much, Dom. I think it was just useful to give our audience just some background on the very important work that you are doing in the malaria area. So with that, I'd like to hand over to Claire, who's going to be our main presenter today for her presentation on its lead optimization with the aid of StarDrop.
All right, so today I'm just going to talk about how we've been using StarDrop to support our program here at the University of Cape Town and H3D. And before I talk about how we've been using StarDrop, I just want to give you a brief introduction of our research context for this particular project. So Dom has introduced it a little bit, but we've been collaborating with MMV since 2009 on malaria projects, and this one is part of this. So as you may know, malaria is caused by a parasite, Plasmodium, of which Falciparum and Vivax are the most deadly strains, and it's transmitted by the female Anopheles mosquito. And these days we still have more than 200 million cases of malaria around the world and more than 400,000 mortalities. So it's still a big burden. The target for 2030 is to reduce the incidence and mortality of the disease by 90% compared to 2015. But we face different challenges, the main one being the emergence of resistance in Southeast Asia and South America. So there is a very big need of new treatment for malaria and transmission blocking. So here we've been focusing on developing oral drugs for the treatment of Plasmodium and Vivax malaria, and we're looking for drugs that are differentiated and not cross-resistant to the current drugs in the clinic. We get our main chemical matter from phenotypic screenings that are coming from pharmaceutical companies such as Biofocus, and we are very lucky to work in collaboration with MMV, who allow the screens to occur. And we also screen some libraries here at UCT. So I'll talk about how we analyze these libraries in a few minutes, and then I'll talk about how we use StarDrop to support the optimization of our hits into early lead and then late lead, to improve metabolic stability, solubility, and also to move away from some specific issues. So starting with the hits, as I mentioned, we get our chemical matter from phenotypic screen, and so far we've been getting our chemical matter from the Biofocus libraries. And in principle, all these libraries are screened against in vitro activity and for ADME assays, and that leads to very big set of data that are then given to us. So this big cloud here on the top left panel, let me get the pointer, is basically what we get when we get the data set, where each point is a compound, so it can be quite overwhelming at the beginning. Like what do we do with that? Where do we start? So what we can do is to apply some filters with some criteria that we are interested in to select the compounds, such as activity. If, for example, if we want in vitro activity less than 1 micromolar, we can say that we want this. We can also set the cytotoxicity above 10 micromolar, for example, because we don't want our compound to be cytotoxic. We can select a threshold for the solubility or for the log-D, and this allows to get a much simpler cloud of compounds that looks more reasonable to be analyzed. So once we've done that, we can look at the structure more closely and cluster the compounds by structure similarities to get some series that have the same chemical structures. And we can also define some scoring profile for hits, applying some criteria that we think are important for a hit, such as the one in the panel at the bottom, with the activity, cytotoxicity, log-D, solubility, for example. And then we can run our different series against that scoring profile to see how the series perform. And if we plot the series against the scoring profile, we get a plot like the one on the bottom left. And we can see that some series don't perform very well in our scoring profile, and we are not likely to work with them. But some of them have quite high scoring profiles, and we may have a look at them and see if they will be interested to be progressed further. So once we've chosen the series based on chemistry and biological data, we can have a look at them closer and see what the strengths and the weaknesses are for each series. So here we have chemical space where each cluster represents a series, and we can color or size the dots by different properties such as activity or solubility to see how the compounds perform in different assays. And if we superimpose different plots, we can have a multi-dimensional view of the series and really have an idea of the strengths and weaknesses of each series. So for example, the one at the top, we can see that the compounds are quite potent for different activities against the parasite in different life stages, and they are not very soluble. So solubility will be a thing to solve if we decide to continue with this series. On this slide, we can see that for some series, if we look at ERG inhibition or cytotoxicity, we are very likely to have difficulties to move away from these activities that are not desired, because it looks like it's more series specific and not compound specific. So this helped us to choose series and to see to evaluate if we would be able to progress the series further without being stuck with one issue that's inherent to the series. One thing that we can also do is if we have series that are similar structurally, we can have a look at which substituents have been used to constitute the library, and see where we may have where we may not have some substituents, like the spaces that have been used and not used, to design further targets later. We can also visualize and establish the initial SAR from the high throughput screening using some function on StarDrop, which is quite useful to start the hit to lead program. So once we're happy with our library and our hits, here in the lab, we're going to resynthesize the compounds and test them to see if the activities are confirmed. And then we'll have to go from hit to lead optimization to see if we can improve the properties of the series. So to do that, one of the things that we can do is to set up an early lead scoring profile with criteria that are a bit higher than the ones for the hits, and see how all hits perform in this early lead scoring profile. So for the series that is shown here, we see that the activity and against sensitive and resistant strains are quite good, but we have issues with metabolic stability, solubility, and cytotoxicity. These three parameters are definitely things that we need to improve to get to the early lead criteria. So we started the early lead optimization by improving metabolic stability. And when we did our assay here, when we screened our compound in our assay here in Cape Town, we had only moderate metabolic stability with only 70% of the compound remaining after 30 minutes. So we wanted to understand where the metabolism occurred to be able to block the metabolism along the way. And by using the P450 module on StarDrop, we can see that metabolism is likely to occur on the right-hand side here. So we decided to put some substituent at this position to block metabolism. And we tested our compounds again in our assay, but unfortunately we had similar metabolic stability. So here the prediction from StarDrop and the result from the measured metabolic stability didn't quite well correlate. But if we have another look at the prediction using different SIPs, we see that metabolism is likely to occur on the left-hand side, which can explain why the metabolic stability wasn't great in our assay. So we decided to block the metabolism on the substituent on the left-hand side as well. And with that, the metabolism is predicted to be very low in the P450 model with CYP3A4 on StarDrop, and it correlates very well with the metabolic stability that we get in our assay that says that it's 100% stable. So once we've sorted the metabolic stability, we applied again our new leads against the scoring profile, and we were pleased to see that the metabolic stability was improved, of course, but also that that blocking the metabolism and adding substituents on at the R1 and R2 position allowed us to improve the cytotoxicity as well. So the main problem that we had to resolve was the solubility. So we had a look at that, and unfortunately the predicted solubility is known to be not very reliable to correlate with the measured solubility. But what we can see is that if we have a high intrinsic predicted solubility, high predicted intrinsic solubility, sorry, we have more chances that to have high solubility in our assay. Also, another parameter that we can look at is in the sp3, the number of sp3 carbons in our compounds, and we see that this correlates a lot better with the measured solubility than the predicted solubility. So if we increase the number of sp3 carbons in our molecules, we are more likely to have high solubility. So we applied that principle to our series, and we made a good number of compounds with saturated substituents to increase the number of sp3 carbons, and we managed to reach high solubilities, and it was very consistent across the series. So here we can see that solubility and number of sp3 carbon correlate very well. So we are very happy with that, and again we applied our new leads against the scoring function, and we can see here that the scoring profile has improved very much compared to the initial scoring profile, and that all the parameters that we selected for our early lead perform quite well with the new leads. The only thing that we can observe is we have a slight drop in potency due to gain solubility, but it's still very acceptable. So once we've done that, we had the early lead and we had to move into the late lead stage, and there we ran into issues that we didn't expect, and unfortunately we had to run away from it. We don't quite understand what's going on, but everyone agreed that it's something that we don't want. So what happened with some compounds is that instead of having a nice sigmoid curve for the activity against the resistance strain K1, we had that weird biphasic bumpy curve here on the right-hand side, and we quite don't understand why we get that second bump here. So to try and move away from this issue, we had a look at different properties to see if we could get any correlation between physical-chemical properties and this behavior against the K1 strain, and we couldn't get any correlation with any of the parameters except the TPSA, which we don't really know why there is a correlation with the TPSA, but that's what we observe. And one interesting thing is that if we had a TPSA of 66.5, we were very likely to have a biphasic dose response. Also, if we have compounds containing nitrile groups, we had biphasic curves. So these relationships are not very well understood, but what we wanted to do was just to find a chemical space where we wouldn't have that biphasic curve, and looking at the TPSA, we managed to design compounds that didn't show that curve, and if we increase the TPSA, we actually move away from the biphasic curve. So once we had that, once we had managed that, we knew which chemical space we had to work in, but we still had to find compounds that were active and that had a good physical-chemical properties to match the criteria for the late lead. So the best substituents were the p-pyrazine-like substituents. So one of the compounds that matched all our criteria was this compound, and it didn't have that biphasic curve. So that's where we stop for the early lead, for the late lead. Sorry. One last thing that I want to show is that we also use StarDrop to look at the progress that has been made for a period of time in the project, how we manage to move away from issues, and the visualization tools that are allowed in StarDrop are very useful to do that. So one example here is how we manage to move away from that K1 dose response and still being in a good chemical space with a good ligand lipophilicity efficiency and no cytotoxicity. So if the x-axis is the time. So if we look at how we've progressed along the time, we see that we had some cytotoxicity issues here and some dose response that were biphasic here, and by identifying a good chemical space where we didn't have the biphasic curve, we also managed to stay away from cytotoxicity and to have good physical-chemical properties. So that's quite useful to show our collaborators the progress that have been made, and yeah, we find it very convenient. So just to conclude on how we use StarDrop, so as I hope I showed that we can quite efficiently pull out series of interest from a big data set, and based on our criteria for activity and physical-chemical properties, we can also easily identify strengths and weaknesses of a series by plotting different properties and using different modules in StarDrop. Using the scoring profile is very useful to rank series and compounds at different stages of the drug discovery development. And then the metabolism prediction are pretty accurate, and it's very useful for our program. And also the way we can look at different trends and look at different correlations, we use it a lot for our programs here, so hopefully people can also use it. So that's about it for me. Thank you very much.
Thank you, Claire. That was an excellent talk. Thank you very much indeed. I'm sure the audience will have many questions for you. Whilst you're doing that, I hope that our audience sort of appreciates that the challenges that organisations like H3D, MMV as not-for-profits and academic groups have in sort of developing novel drug candidates in a really very challenging area. These areas that they're working on are typically in the neglected and developing world diseases or even niche indications. And discovering new drugs in these sectors is even more challenging than in pharma that we sort of face on a day-to-day basis. And there are a number of reasons for this. Obviously, the diseases themselves are very challenging. And often, of course, they're affecting patients in the developing world. And of course, in the developing world, there's also an emerging resistance to the current therapies that are available to them. Now, the groups themselves are also sort of faced with the challenge of limited resources and the need to sort of manage that complex data that we all have to work with. And often they're generating data across large sort of multi-center collaborations. Now, I do sort of appreciate that the way that we work within large pharma and even within biotech is also changing and evolving currently, and that it's becoming more collaborative in nature, where we're working across interdisciplinary groups, for example. We collaborate with CROs and academic groups, for example. And so there are sort of parallels in the challenges that we face. So whilst you're sort of popping your questions in for today's speakers, I just would have wanted to sort of pose to our panel how they're currently sort of tackling these issues with working from data from different sites, and how they're sort of using different tools to actually make the best decisions with the data that they actually have available to them. So first thing I'm going to do is I'm just going to unmute everybody so that everyone can speak.
Yes. So I may start, maybe, Dominique speaking there. I'll start from the angle of MMV. So definitely on our side, this is a key topics to be managed. How to deal with a network of persons, scientists across the world around a single object, which is a project or a molecule, is something that is quite challenging. So there are several axes to do that. One of the axes is obviously to have everybody accessing the information. So we have databases that have now been developed and that are quite convenient for project-based organization. But definitely another point is how do we take the decision on how to move, how to select the best compounds and what to do in the next steps. So in that case, having an integrated tool like we have with Optibrium, with StarDrop, is something that is great because people are discussing on the same object at the same time, which is absolutely great when you work with academic partners that need to have access to those information or those tools to progress in the right way their environment. So communication is certainly a key point, and sharing of information using typically a tool like StarDrop is something critical.
I think Dom makes some very, very good points. I think that ability to collaborate and to share data very quickly and easily is very important, as well as the ability to quickly focus in on the highest quality chemistry, as Claire has just demonstrated from an early stage. That's the way that projects make the most progress. So, for example, we're very happy to be working with UCT and MMV and a wide range of other not-for-profit organizations. A couple of ways that we at Optibrium have done that. Working with Dom and his colleagues in MMV, we've recently created a hit qualification profile, which we'll soon be publishing on our website for any StarDrop user, that really embodies the sort of hit qualification criteria that MMV like to look at when deciding on new series to progress. And that means that anyone working in the anti-malarial space can quickly apply that and see how their compounds stack up against those requirements, any particular shortcomings or any particular strengths that a series or compound may have. The other thing that's really important is being able to get at your data very easily. We're very pleased to work with partners who work in the sort of cloud-based database and collaboration software. So organizations like Collaborative Drug Discovery and also Science Cloud. And again, another collaboration we had with MMV was to link StarDrop directly with those sources of data. So that these multidisciplinary, multi-center collaborations where biology data may be generated in one center, ADME data in another, activity data in a third, all that data is brought together in a single point, and projects like the one that Claire has just described can access all of that data, bring it directly into StarDrop, and very quickly be able to perform the sorts of analyses that you've seen. So these are some really important areas that I think help to address these challenges.
Thanks very much, Dom and Matt, for your insight there. And I guess we're all sort of very aware that sort of technology is evolving and trying to make this process more streamlined and easier for users. We're sort of seeing new developments almost on a sort of weekly basis. I don't know if Matt or maybe Dom would like to add anything further on that before we progress on to the questions from our audience.
Maybe, Matt, you can start, but I have one point which is quite important. It's always quite difficult when you do analysis. You need to to find the right balance between harmonization in the way you analyze the data and flexibility, because sometimes you need some flexibility in the middle to make sure that you are not losing or dropping stuff too early in the process that maybe could be solved later on. So definitely technology should be able to evolve in order to facilitate this, to find the right balance for a project between harmonization, making sure that every project is managed in the same way, and a kind of flexibility in the way you analyze.
Yes, I absolutely agree, Dom. I think it's really important. One thing we're very keen on is not setting sort of hard filters or hard criteria for selection of compounds. There's no such thing as a sort of a black and white decision.
Mm-hmm.
And so, for example, within the sort of scoring profiles that Claire illustrated, you don't have to have just a sort of a hard threshold cutoff, but actually a sort of a soft boundary where, being in a certain range is ideal, but when you leave that range, instead of simply throwing away a compound or filtering it out, you give it an appropriate sort of penalty that sort of gradually changes as you move away from that ideal range. So that if the compound was good for everything else, but not so good for maybe one property, it's still going to rise to the top, and you're not just going to miss that opportunity. Similarly, the ability to take the uncertainty in this data into account. It's absolutely critical to remember that all the data we deal with in drug discovery has a lot of uncertainty. All the assays that we run have a lot of variability, and certainly if you're using calculated properties, there's a lot of statistical error and uncertainty in that. And the last thing you want to do is to throw away a potentially good compound due to an inaccurate measurement or prediction. And so another thing that we do when calculating these scores, as Claire showed, is actually to take that uncertainty into account. So you don't overweight an uncertain value or a value that is within sort of statistical error bars of the criteria that you're setting. So that's really important. And the third is actually, again, as you say, Dom, really important is the ability to change these criteria. While you can have a standard profile that reflects that, for example, the criteria for selection of compounds for an anti-malarial, perhaps if you're willing to be flexible, you can simply change those and see the impact that has on the chemistry you'd select. Does it light up a new chemical series that are worth considering? There's no, as I said, there's no sort of hard and fast one-size-fits-all profile. And so provided you're clear about why you're changing the criteria, it's absolutely appropriate to be able to edit that to your specific project requirements. And again, you can very objectively prioritize your compounds against those modified criteria. So that flexibility is important, particularly in such an important area such as anti-malarials, where the last thing we want to do is to miss the opportunity to find a good new drug.
Thanks very much. That's been very interesting to hear your thoughts on that. I am aware that we've got a number of questions, so I'd like to sort of give our panel an opportunity to answer some of those. So the first one, I think, Matt, you might be in the firing line for this one, is what are the axes of the chemical space plots? I appreciate, Claire, you generated those nice plots, but I'm guessing that Matt would be itching to dive in on that one.
I don't think it's fair to ask Claire for the technical details behind the methods that we apply. So the chemical space plots that we generate, the default is to use a 2D path-based similarity and a Tanimoto index, but in fact you can define these in terms of any set of descriptors that you want, that you think is relevant. But I believe that on the plots that Claire showed, that was just a standard sort of structural similarity. The actual axes don't have a physical meaning. They're combinations of similarities between all of the compounds in the set that gives you that sort of visual clustering to group together compounds in the best possible way. So the actual absolute positions don't have a meaning. What really is important is the distance between points. If 2 points are close together, they are structurally similar, whereas if they're far apart, then they are structurally different. And I'm just trying to see if I can make myself the presenter so I can just sort of illustrate that very, very quickly for you. So I had just, while we've been speaking, popped up a chemical space plot here to address this. And essentially what you see is that these groups of compounds represent structurally very similar classes. So if you point at one of these, you can see the structure. If you point at 2 points that are close together, they are structurally similar. Whereas if you look at 2 points that are far apart, they are structurally diverse on the scale of this sort of example library that I just quickly brought up. So I hope that addresses your question.
Thanks, Matt. The second question is relating to the biphasic dose response that Claire talked about. And the question is whether we think that's related to the solubility of the compounds.
No, we don't think so, because we've got soluble compounds that have biphasic curves and insoluble compounds that don't have biphasic curves. So there is no correlation between the curve that's observed and the solubility.
Thanks for that, Claire. The second question is sort of similarly related, and it asked if we generated our own solubility models based on the solubility data. So did we use, I think the question is relating to the automodeler capability within StarDrop to actually build and validate robust QSAR models based on in-house data and in-house compounds?
I'm afraid we haven't tried that. I'm not sure how we do that actually.
So that's probably something for us to follow up on after the meeting, Claire. we can work with you on that.
That would be great.
Okay.
I would comment actually that on the graph that you showed, Claire. I mean, it's a nice illustration, but I think there's an important point to note there. That the first is that the solubility model in StarDrop, or the one that you were plotting there, is an intrinsic aqueous solubility model, whereas the actual experimental data was solubility in phosphate buffered solution at pH 6.5, and actually physicochemically they're quite different properties, so one wouldn't necessarily expect a perfect correlation there. And actually, given the sort of lower limit of sensitivity of the assay, just eyeballing that graph, it actually was trending quite nicely. So it was actually quite nice to see that there was a reasonably good correlation given those limitations, although it wasn't perfect by any means. And it was nice to see that the fraction of sp3 gave you that additional steer. But we'd certainly be happy to work with you to build a local model of your specific data and those specific conditions, which may give you a much stronger correlation.
Oh yeah, that would be awesome.
The next question is, how were the ADME properties of the HTS library assessed? And was this done using in silico calculations?
No, these ones were measured with the, for the compounds, they also screen the ADME properties, like solubility and metabolic stability.
Thanks very much for the clarification. Next question is thanks, Claire, for an insightful presentation. Just a curious question. Is it possible to do multivariate analysis on StarDrop to identify points of similarities and divergence in the series? Or does this have to be done in a different platform?
I wonder if that might be a more appropriate question for me to address in terms of being the functionality of StarDrop itself. So yes, you can do a wide range of different sort of multivariate analysis in StarDrop. Obviously, Claire has touched on this sort of multi-parameter probabilistic scoring approach to actually bringing together all of this data and find the sort of consistent strengths and weaknesses within a chemical series or an individual compound. So that's the sort of one approach there. The sort of chemical space that we've seen relates particularly to, as we've seen so far, structural similarity, but one can also perform, for example, principal component analysis or other clustering approaches based on chemical properties or calculated properties or experimental data as well. And then there are other appropriate ways that you can sort of easily compare between different series within StarDrop as well. So just to show you one visual way of doing that, I'm just going to pop into something we call card view here. Again, Claire showed some nice examples of these cards which sort of summarize the key properties for individual compounds. But one of the things one can actually do here as well is to group these either by clustering. So you could just run a clustering algorithm here and cluster by common substructure to group together those compounds, for example. So that you can begin to group these together in these sort of structurally similar classes. So for example, this is now a stack of cards. It contains 15 cards, all of which share this same common substructure. And if you look, you can see the individual cards within that stack. You can obviously use this to very quickly generate chemical series, stacks that represent chemical series. So here you can see sort of 3 clusters that belong together, which I can group together into a chemical series. Or just to save a little bit of time, I will just stack all of these by a label for the different chemical series. So you can see there are 6 different series within this data set. And now what's sort of nice is, although right now we're just showing structural information on here, what I can actually do is begin to look at how the properties of these series compare. So on these stacks, as well as structural information, I could, for example, put information about distributions of other properties. So you could compare, for example, log P, the molecular weight. It could be experimental data or calculated, or look at solubility, for example. And then you can easily sort of just scan across these and see which of the series are having, this has a low molecular weight, low log P, moderate molecular weight, and high solubility, which looks pretty good, while some of these series here are getting sort of high log P, high molecular weight, and moderate solubility is perhaps not so good. So very quickly and easily visually compare between those as well. One can also group compounds into stacks by properties and many other things as well. But that just gives you a few sort of illustrations of how one can look at this sort of multivariate analysis.
Thanks, Matt. This one is almost certainly for Claire, but can you touch on your approaches to select hits from the HTS? Did you use priority in terms of the parameters that were used? Did you use clustering or maybe other options?
For the initial library, we first use filters to simplify the data set. So we set up the activity for in vitro for the asexual blood stage to 1 micromolar, and then the cytotox to more than 10 micromolar, and the solubility. We put a threshold of 50 because we don't want to eliminate all the compounds that are not soluble, but we don't want compounds that are completely insoluble. And also for the log D, we were a bit more relaxed for the filters. And then for the criteria that we use for the scoring profile, we apply, we made the scoring profile ourselves, but I think it must be quite similar to the ones that Dom was talking about for MV. And I don't know if I'm answering your question.
No, I think you have that. Thank you very much, Claire. Yeah, so I believe that early on in the project, requirements in terms of the properties required for the compounds were shared between MMV and UCT. And I think this is some of the work that Matt sort of briefly touched on earlier on, where we're going to make these profiles available to the wider audience as part of the Optibrim community website.
Yeah.
So the final question that's just come in is related to how we generate the compound structures within StarDrop and whether we use SMILE strings or whether we have to use other sources. But the person posing the question has actually answered the question themselves correctly, and that we can use SMILE strings. StarDrop itself is very flexible in terms of file formats that it can work with as a starting point.
Yes, I think it's worth saying that obviously SMILES is one standard format. We can also read SD files, MOL2 files, all the usual sorts of formats. And of course, StarDrop can be connected up with various databases. So if you have a database that will accept a CAS number and return a structure, then certainly StarDrop could query directly into that. So yes, there's many different ways one can bring structural data into StarDrop.
Thanks, Matt. So those are all the questions that we've received so far. Before people start to sort of leave the meeting, I would like to thank Claire once again for an exceptional talk. It was really very interesting, and thank you so much for putting so much time into preparing this talk for us. And thank you again, everybody, for turning up today.